Hi, this is Dr. Launen, and this is a short lecture on hemoglobin and sickle cell anemia and sickle cell disease as a case study for proteins and genes in genetics. Hemoglobin is an essential transport protein that is located in our red blood cells. Its job is to pick up oxygen when the red blood cells are up near the lungs, the highly oxygenated area of the environment, carry that oxygen deep into the tissues, where oxygen is continually being used up, therefore creating a concentration gradient, and that allows the oxygen to detach from hemoglobin and to passively move across these cell membranes and into the tissues. In the same areas, CO2 is found at higher concentration in the cells outside and around the red blood cells, and so CO2 will passively diffuse into the red blood cells, and it will be carried by the red blood cells back up to the lung areas, and then that CO2 will be transported through passive diffusion out into the lungs and be exhaled. So hemoglobin is an essential oxygen transport protein in our blood. It's also an example of a protein that has quaternary structure, meaning more than one peptide makes up the total protein, the total functional protein. Each hemoglobin molecule is made of 4 subunits, shown in different colors here. Each subunit is its own individual peptide that is folded to assume three-dimensional structure as shown here. A feature of hemoglobin to note is that each subunit contains a heme group here, 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 and here. The heme group looks like this. So this is not a protein, it's a structure, it's an organic molecule, and it's actually the structure that binds oxygen. These 2 subunits are essentially the same in adults. They're the beta genes or beta subunits, and these 2 subunits are essentially the same in adults. They're the alpha subunits. Let's consider the genes that have to be expressed in order to build a hemoglobin protein. In adult hemoglobin, we use this beta gene twice in order to get the 2 beta subunits, and that gene is located on chromosome 11. Also in adults, we'll express these alpha genes, and we'll express both of them in order to produce 2 alpha subunits, but functionally these genes are so identical that you can almost think of them as one gene. That's the adult scenario. When you are an embryo or a developing fetus, you will build your hemoglobin somewhat differently. So embryos will express the epsilon gene instead of the beta gene in order to build the equivalent of beta subunits. Fetuses will express a combination of these gamma genes in order to, this beta gene, in order to build the beta equivalent subunits. And then delta is expressed a little bit in all humans at different stages of the life cycle, and I won't really get into it here. Early on in development in the embryonic stage, instead of utilizing these alpha genes, the embryo will utilize these zeta genes in order to build the alpha equivalent subunits. So over time, basically as we move from 5' to 3' on these chromosomes, and 5' to 3' down here, we'll sequentially express these, and then a combination of these, and then these genes as we make adult hemoglobin, which is fully implemented about 18 weeks after birth. All right, so let's think about when things go wrong. So normally, focusing in on the beta gene, this gene here, okay, we're going to focus on that for the rest of this talk. That gene has a particular region in the gene that encodes glutamic acid, a particular electrically charged amino acid at position 6 in the peptide. This is the N-terminus, okay? So it's a position very close to the N-terminus. Now, normally, in the HBA, or what's also called beta subscript A allele for the beta gene, you get a DNA sequence that encodes glutamic acid. However, in many individuals, there has been a mutation event, and it changes the DNA sequence that encodes this amino acid and into something different. And as a result, you get a different amino acid at that position. It's a sense mutation. And that new mutation, or the mutant form, contains the amino acid valine, which is electrically neutral. It's a nonpolar amino acid. So let's think about this a little bit. We're going to move on to talk about a disease that's called sickle cell anemia or sickle cell disease, SCA or SCD. If you have this mutant allele in one copy, in other words, you are heterozygous, then you have a condition called sickle cell anemia, and you might live a fairly long and reasonably normal life. If you have two copies, you're homozygous for this mutant allele of the beta gene, then you will have what's called sickle cell disease, and you won't live to be very old and you will be extremely ill. This is a genetic condition that has existed for thousands of years, and but it was first documented in 1910 when Walter Clement Noel, who was a dentistry student studying dentistry in Chicago as a visiting student from Grenada, the island of Grenada. He wasn't feeling well, he had pain, and he had other symptoms of anemia, and he went to see physician James Herrick, who wasn't actually all that interested in his condition, and he punted to Ernest Irons, who actually examined Noel's blood under the microscope and saw sickle-shaped cells. I'll show you a picture of them in a moment. And so he first described this condition, and then people started to study it, including Linus Pauling. But the person who first published and clearly showed that this genetic condition of sickled red blood cells, which is associated with anemia, pain, swelling, and other phenotypic aspects, the first person to publish this as a genetic condition was E.A. Beat, who was a military doctor working in Mozambique back in 1947. Then in 1949, Beat again, and separately James Neal, published further papers documenting the inheritance of this condition. And by that, I mean they documented the fact that the conditions were associated with a mutant allele for hemoglobin, and that in heterozygotes you had sickle cell anemia, and in homozygotes for the mutant allele you had sickle cell disease. So let's take a look at the phenotype at the level of the protein. And so first I'm going to point out this is a different way to write the genotype. You can write the genotype of a normal person who does not have sickle cell disease as beta A, okay? Or you could also write A, and you'll see, different traditions expressing that same genotype. But this is a normal individual. When you take that normal individual and you extract hemoglobin from their blood, and you study just the beta subunit, okay? You get one band because that normal individual is only making this form of the beta subunit, all identical, one band by gel electrophoresis. Similarly, actually, if you extract red blood cells from a person who has sickle cell disease, so they're very, very sick people, you're going to be doing this at a young age because they won't survive to old age. They have both copies of this mutant allele, and they make, therefore, only one mutant form of the beta subunit. And so it will run to a different place by gel electrophoresis because it's chemically different, but it's all one protein. If you extract if you extract hemoglobin from the red blood cells of people with sickle cell anemia, these are the carriers for the mutant allele, and you focus on the beta subunit, you'll see two forms of the beta subunit. One is formed when they express their normal copy, and one is formed when they express their mutant copy of the beta gene. And that's sickle cell trait or sickle cell anemia versus sickle cell disease. This is an example of codominance because at the protein level, you see both phenotypes, both forms of the beta subunit present. So looking at this, here is the double-stranded DNA, right? This is the normal form at that position, the normal codon that encodes glutamic acid. This is the DNA mutation that gives rise to a different messenger RNA codon and therefore valine as the amino acid that's in that particular position. And then at the protein level, we thought about that, what it looks like on the gel, but I think it's more important to look at these pictures because these show what an example of what a normal hemoglobin protein would be. 1, 2, 3, 4 subunits. There's the hemoglobin protein. It's not stuck to any other hemoglobin proteins, which is good because it allows the red blood cell to function normally in that case. In contrast, if you have two mutant hemoglobins, and that means they have two mutant beta subunits, or even one mutant beta subunit, then what will happen when they bump up against each other is they'll stick together, and you get these strings of clumped hemoglobin, and that causes the whole red blood cell to sickle or take on this abnormal and unhealthy shape. And those sickled red blood cells, when they get into areas where they're trying to squeeze through little tiny capillaries, they clump and they block, and then you get edema or swelling in the affected areas, and you don't get oxygen transport. So you have a lot of really terrible cardiovascular issues. And that's different from the normal healthy individual that has normal hemoglobin, which does not clump and allows the cells to keep their jelly donut shape, and they flow beautifully through the tissues. If you've got sickle cell anemia, so you're heterozygous for this, then what happens is most of the time you've got healthy looking red blood cells, but under extreme stress, like pregnancy, high altitude or extreme oxygen demand through like long distance extreme aerobic activity, then the cells will start to sickle. And if you look at the blood, you'll see a mixture of sickled and unsickled cells. If you've got sickle cell disease, you only have the sickled cell. And this picture doesn't actually show that. It shows a mixture. So if you had sickle cell disease, they would always sickle when they were in aerobic areas and therefore clump throughout the body. All right, so there are many known hemoglobin mutations, and we just talked about this one right here, okay? And it's a mutation that affects the beta chain of hemoglobin. This is the normal sequence. This is the sequence that we just continued. There's that problematic valine. There are many other mutations that can affect the beta subunit of hemoglobin, and there are also mutations that can affect and change the alpha subunit. And all of these are associated with different levels of disease and occur at different incidences in population. I want to point out one more thing before I close this lecture. And to do that, I'm going to go back up to this genetics area, and I'm going to point out that, interestingly, while most of us have predominantly adult hemoglobin in our blood, we do all produce a little bit of hemoglobin that contains this fetal beta subunit, about 1% in the average person. So these genes, when I tell you that they're turned off entirely as we transition from embryos to fetuses to babies and then adults, that's an exaggeration. There is a little bit of leaky expression, enough to make about 1% of our blood contain fetal hemoglobin, and not just adult hemoglobin, but the dominant amount is adult hemoglobin. And with that, I will close this case study video lecture and have a great rest of the day.